Welcome everyone. My name is Robyn Ball, and today I'm going to talk about some tools that we built at the Jackson Laboratory to really help us traverse the mouse-human interface, and particularly for human disease. I'm a data scientist at the Jackson Lab. So just a quick overview. I'm going to give you some motivation of why do we want to do this, right? And then I'm going to talk about the tool suite. I'll give you some examples after that, and then at the end of the slide, I'll have some resources with QR codes. So if you want to take a picture of that, that'd be great. So why do we do this, right? So we study mice to help people, right? And unfortunately, we do these like large human GWAS studies, and we find some variants that confer a risk, but we don't know much more than that, right? It's very hard. We don't have the power to characterize those variants and those genes. So it often leaves us asking, well, how does this work? Where does it work? How does this influence the disease process? And so we can use the mouse studies to do this. We can use it to characterize and interpret these genes and variants. So our solution is really to integrate these data resources that we have at Jackson elsewhere, so that we can, if we find human variants and genes that can be targeted, then we can also look at the mouse and say, well, can we use these mice to study for preclinical studies, and how? Where is their shared information? Alternatively, if we find something in the humans, then we can also go back to the mouse, or we find something in a mouse and say, where does it interact with the human? So when everything's connected, right, we can use this information, this knowledge graph, to do this work. And it allows us to ask a lot of different questions. For example, if we're going to study some disease-related traits in humans, which mouse traits correlate to that? Which mouse traits should we actually study? What variants influence complex disease traits? Is this a drug-interacting gene, right? Once we find is it drug-interacting, which variants and genes have shared roles across species, etc. So the ones that I'm going to talk about today are really, what variants influence complex disease, and do they have shared roles across species? How can we find that out? And then how can I also programmatically retrieve this information, programmatically retrieve disease-relevant gene sets? So in order to answer this question, we need to know what variants in the mouse influence disease, right? Before we can get to where it's shared across species. And so for that, we've built out these data resources and these analytical services. We're going to need a saturated grid of genomic information in the mouse. We're going to need data integration across mouse traits and across mouse populations. And then we're going to need to be able to map this information across both mouse and humans. So the first tool I'm going to talk to you about today is Genome Muster. So Genome Muster was recently published this year, and it's a saturated grid of mouse genetic data. And so it has 657 inbred strains at 106.8 million sites. So it's a large and unified dense grid. So why did we build this, right? The challenge, as many of us know, is that there's very sparse data for most of these strains. And if you try to integrate across data sets, it doesn't really work well. And so if you want to use this data in analyses, often you have to exclude strains or exclude large regions, and that really hampers us to find what we're looking for. So this figure on the circles plot on the right here, Genome Muster density is on the outer ring, so you can see that's what it's relative to. And then all the data sets that went into Genome Muster and these other rings. The largest one is the Sanger data set. It had 53 strains and about 80 million sites. So this is still only about 75% of what we get in Genome Muster, and less than 10% of the strains represented. So we had to have a very accurate imputation algorithm to start building in all of these gaps across these strains. So what we did is and I can talk to you about this after, I don't have time to go into the details, but we developed this imputation algorithm that is quite fast and also quite accurate. And so what you're seeing here on the right is a histogram of each strain-specific accuracy. So how did we do that? Well, in each region, we randomly sampled some data. We left it out of the imputation procedure for each strain, right? We ran the imputation, then we can compare that strain's predictions to the actual data. So that's how we estimate this accuracy, and we do this across the entire genome for each strain. The nice thing about this is you get a local confidence level, right? So if you're not comfortable with that level of accuracy, you can filter it out, or you can use it to weight your downstream analyses. And you can access this at muster.jax.org. So now I'm going to translate. Okay, so now we have all the genomic information we need. We also need the trait data, right? And the mouse phenome database is what gives us this information. So there's about 28,000 trait measures that's been collected over these decades, and a large number of mouse strains. So we can use this data in the GWAS meta-analysis, which is integrated in the mouse phenome database with Genome Muster. So this allows us one tool to do a lot of different types of analyses. And then the next thing we need, right, is being able to map this information across species. And so we've built the variant graph, which uses regulatory information to really make these relationships. It's not just the closest gene, right? It's all this information about EQTL and other things that we've built into the graph. And so not only do we have the variants and the genes and the transcript relationships, but we also have the regulatory peaks in there, like H3K-ME3, for example, right? So you can really like fine tune what you're looking for. You can filter on the tissues that you're most interested in, et cetera. There's lots of different ways you can filter this data, but it's a large graph. It's built in Neo4j, and it enables us to do this work quickly. So now I'm going to talk about examples, right? I've given you kind of a whirlwind of the tools, right? So I want to show you how do we use that really to make discoveries. So I'm going to show you two different examples. In the first one, what if we just want to, forget about other things? We want to find genes associated with disease using mouse data, right? How do we do that? And then if we also have human data, how can we interpret the data from the humans where we have this shared signal, this, shared roles across both mice and humans? So direction one, finding disease-associated genes using genetic meta-analysis in mice. So like I said, we start here with the mouse phenome database and all of the different trait measures, and then we use the genomic information in Genome Muster to do GWAS meta-analysis. Once we have the meta-analysis results, we filter them for significant effects, and then we use the variant graph to map these variants over to mouse genes and their orthologous human genes. So in this example, we were looking at diabetes. So we have a lot of diabetes-related traits in the mouse phenome database. We have these data have been expertly curated, so it makes it really easy to group them based on ontology terms. So for example, I'm giving an example here for blood glucose amount. So all the measures that have been annotated to blood glucose amount, we can easily just pull that in and use it in a meta-analysis. And there's lots of other information, right? Not just the measurement type, like glucose tolerance test or bone density, but we know which diets these mice were given, whether it was a high-fat diet or a high-fat, high-sucrose diet, any treatments that they received, and then we can do this across all the populations because now we have Genome Muster, right? We're not limited to one small set of strains. So what does this look like? So first, I just looked at glucose, right? Like if I found all the traits related to glucose, we can do a meta-analysis. And then we looked at all of the traits where the mice got a high-fat diet. So this is obviously a very heterogeneous set. There's lots of different studies involving a high-fat diet, whereas glucose is much more diabetes-related. And so what I did was I mapped the variants that we found through the variant graph to the genes and then to the orthologous human genes. And then I went over to Alliance and I said, okay, well, what diseases have these genes been annotated to? And what you see in the top right here is that for the glucose meta-analysis, type 2 diabetes came up as the top hit, right? Most of the genes mapped to that. And then we had Alzheimer's disease, obesity, and some other diseases behind that. Whereas high-fat diet down here is, of course, it's much more heterogeneous. It affects many parts of the body. And so we see like Alzheimer's disease is the number one, and then lots of other inflammatory diseases below that. And so that was a really interesting result. So we can see that we actually are getting signal from these data, right, that map onto the human people with the disease. So the direction two that I'm going to talk about is really, okay, let's say we have human GWAS on the right and we have mouse GWAS on the left. How do we find these shared effects? And then can we characterize those variants and those genes that have orthologous targets? And so that's the idea here. So here on the left, we have lots of addiction-related data in the mouse, and then also addiction-related data in the human. And this is a really kind of complex figure, but let me boil it down for you. So we have my colleague, Alexander Hatoum, at the PGC has done a lot of meta-analyses related to multi-substance use. And so he and his collaborators have identified a set of genes, right, related to multi-substance use in people. So then over here at the mouse, I said, hey, we've got some of that same data, right? We've got a lot of data in MPD. So we have this ontology term, response to addictive substance, which is this blue ring here. And then we have cocaine, which is this green ring, alcohol, nicotine, and then also the withdrawal response, right? So we can do these different meta-analyses across all these different substances. And the Fuji plot here, which is what you're seeing, is really gives us a bird's eye view, right? It's very hard to look at this much data. So we can take a bird's eye view of all of the work and we can start looking for signal. So I'll just note that the genes here from the variant graph are annotated to each SNP. We're only considering significant SNPs, and then here it's better if you look over here. So then we can quickly see for these SNPs, like some of them look to be quite specific to a certain drug, maybe cocaine, whereas other ones are shared across these different substances. And indeed, we did find some genes that were also found in the human studies. The human studies is a very small set, right? So we found much more in the mouse, but we can look at the ones where we find concordance. But the thing is like, okay, so we find those genes, that's great, but what does it actually mean, right? In the human, we do these studies and they say, okay, we found this gene, PDE4B. We don't really know what it does just from the meta-analysis alone. So can we use the mouse to really understand what that looks like? And indeed we can. Right, so we start with mouse GWAS and human GWAS. We go through all of this to find the shared signal here in the middle, and then from there we can go back to the mouse data, right? We can go back to those genes and variants and traits that went into that meta-analysis. And that's what we're seeing here. So we took those genes like PDE4B, and we said, okay, what traits is that those variants affect in the mouse? And they're all withdrawal-related traits. And this is supported by other literature, and it's actually being developed right now, is that PDE4B is a good drug target because it's related to withdrawal of multiple substances. So imagine if you could influence that gene, right, so that the person doesn't necessarily feel that withdrawal effect as strongly, maybe it would help them get off the substance. So that's the idea, and I know I'm talking kind of fast. But we only have about 5 more minutes. So I'm just going to show you some of the tools now. So this is an illustration of the variant graph UI. So it's in Neo4j, as I mentioned, and so we can search the variant graph through the UI and return the results. This is an example of studying inserting PDE4B. So PDE4B in the mouse is here. This is not shown, but there's lots of variants in the human that it maps over to here. So we can get all this information. We can find all those variants through the variant graph. Now, GeneWeaver also we can use here, right? So I haven't talked as much about GeneWeaver, but GeneWeaver has about, GeneWeaver has about 200,000 gene sets, right? And these contain lots of different studies. Some that are published, some that are different, and you can filter them based on, how, if it was curated by an expert, if it was published, etc. If it's just something someone uploaded, you can upload your own gene sets and do analysis using all the other gene sets. So it has a lot of information in there. There's many, many different analyses you can do. One that is of particular interest to me right now is, can I programmatically retrieve all of the gene sets associated with a disease or associated with a set of traits that I'm studying. I don't want to go one by one, right? I just want to send to say, can I just get everything back? And so Alexander Berger at the Jackson Lab has developed this new search API, which is accessible to you. You can get it at this QR code. And it allows us to search for a disease term or phenotype term. It uses natural language processing. You can also do a match based on ontology IDs. So whether that's disease ontology, human phenotype ontology, mammalian phenotype ontology, etc. You can also match on PubMed IDs. And then you can then filter, right? You can bring in all this information. You say, well, I really want data with only continuous results. I don't want just 0 or 1. It was in there or it wasn't. So then you can filter on that. You can filter on the type of tier that it was curated under, whether it was expert curators or not. And then you can just automatically download everything and use it in your analysis. So this is just an example of using the API where we're looking for all of the gene sets associated with addiction, but not alcohol, right? So you can use standard ways of searching these data that you would in a web page. And then here's just those that were annotated to a mammalian phenotype term for addiction. This is just an example of what you get back. So I know I'm running out of time. So just some acknowledgements. I've worked very closely with Matthew Gehring, Alex Berger, Molly Bogue, Hongping Liang, Dave Walton, Vivek Phillip, Eric Baker, and Alyssa Chesler at the Jackson Lab to build these tools, among many others. This work would not have been possible without our funding agencies through the NIH and through internal funds from the Jackson Laboratory. And as promised, here are some QR codes if you want to use those to get to our resources or take a picture so you can get to them. So, do we have any time for questions?
A couple minutes.
Great. I know I talked real fast because I wanted to hear your questions. Was it too fast?
Can you use the microphone in front? Just for the online audience. Thanks.
That was a lovely talk, and I'm wondering whether there are diseases for which mouse models do not represent accurately the human disease, because as you know, the complexity is going to be different. The molecular mass is going to be different, which affects the networks.
Yes, that's absolutely true. One of the reasons why that is simply because we're choosing the most convenient mouse often, right? There's the Black 6 mouse, is what we've mostly characterized. It's just the mouse we choose. But in reality, we have 14,000 mouse strains to choose from, right? So that's part of it, is that we want to identify not just the traits that are going to go across to the human, but also the particular mouse strains to use in our studies that are going to be most susceptible and have that phenotype that we're seeing in people. Great question. Anything else? Okay.
Okay, do you want to help the next setup? And I'll take the initiative to ask a question as well, since we have a minute left. And I'm just wondering, you're able to map between human and mouse common gene set. Is there a way to extend beyond that and look at common pathways?
Yes, sorry, I was going to get over to the microphone. Yes, absolutely. So that's what we use GeneWeaver for, because we have all of that data in GeneWeaver. But that's something we're starting to build in now more and more. Yes. So I'll get out of your way. So thank you.